Hello everybody, welcome back to Creation Myths. The video you are about to watch is a rebroadcast of a conversation I had with Rebekah. That's Rebekah of the Bread of Life channel. This is a conversation we had over on her Examining Evolution channel. The original video is linked down below, so if you don't subscribe to that, go do that. In this conversation, we talked about endogenous retroviruses and how they are evidence for evolution. I think it was a really good conversation. I had a lot of fun with it. So here it is. I hope you enjoy. Thanks for watching.
Hello, everyone. Welcome to Examining Evolution. I'm here with Dr. Dan Stern-Cardinal, and he has a YouTube channel called Creation Myths, where he just kind of works on the opposite side that I'm doing. But I like to hear both perspectives. Dr. Dan is an evolutionary biologist, and he's going to share with us today the evidence for evolution from endogenous retroviruses. So thank you for being here, Dr. Dan.
Yeah, happy to. Thank you for having me on. This is my first time on this channel, but it's my second time talking to you because I was on your other channel one time.
Yeah.
And that was a lovely conversation. So I'm happy to be here. It's always nice to talk to you. And we got a fun topic. I like herbs a lot, so this will be fun.
Okay, cool. Well, do you want to get us started in what you want us to understand about how ERVs are, evidence for evolution?
Yeah, so the big idea. So first, herbs, what the heck? So it's ERV, which stands for Endogenous Retrovirus. So these are basically the relics of viral genomes. There are types of viruses, retroviruses. One of the things they can do in their life cycles is they integrate into the host DNA. So they can cut a little hole and stick themselves right in there. And if they do that in germline cells that are going to propagate to become either egg or sperm, what can happen is they can get stuck in the genome. So they get embedded in the germline. Those germline cells become sperm cells. The sperm fertilizes an egg. And now that endogenous retrovirus is going to be that viral sequence is now going to be present in every cell in the next generation, right? So zygote's going to have it, and everything that grows from that zygote is going to have that viral sequence in its DNA. So that process is called endogenization, right? So they get stuck in the genome. So these endogenous retroviruses, what often happens is when they do that, when they endogenize, they mutate. And it's actually one of our, the mammals do this, is we seek out these sequences and mutate them so that they can't activate and cause problems as actual viruses. So they get stuck in your genome, and then they degrade over time because there's nothing keeping them there. So we can see for humans, it's like 200,000 sequences that were originally from retroviruses that incorporated into our genome. It makes up about 8% of our DNA. Of these, only a handful, like 15 to 20 of them, are what we call complete herbs, where they have all of kind of the components of the actual virus genome. And the rest are missing something. It can be just missing one little piece. It could be missing almost the entire structure, but there's little telltales that we can always recognize and be like, aha, that thing has this little bit that we only see in retroviruses. So that's a retrovirus derived sequence. So we have these retroviruses in our genome. And in terms of evolutionary biology, the really, really interesting thing is that they provide this beautiful roadmap for common ancestry, because we can look across, big groups of organisms. I really prefer to consider mammals, right? Just all mammals. You can look at the pattern of shared retroviruses across all of the mammals, and it falls out in this nice, what's called a nested hierarchy, which for anyone who's not familiar with that term, you could think of a nested hierarchy as a box within a box system where you have small groups inside of increasingly large groups. You get your species, genus, family, and so on, all the way up for mammals to class Mammalia is kind of the taxonomic level. So you have, within the smallest boxes, you tend to share the most endogenous retroviruses. As the boxes get bigger and bigger, you share fewer and fewer. So we have what's called this nested hierarchical pattern of sharing ERVs, and that is just a beautiful illustration of kind of the process of evolution taking place over time and leaving its telltale signs in these genomes of all these organisms, all these mammals that shared common ancestry, millions and millions of years in the past. That's like the 30,000 foot view version of what ERVs are and why they matter when we're talking about evolution and common ancestry. Oh, Rebekah, you're muted.
Sorry about that. Okay, so
I do that all the time.
I guess my first question about that is, how would we know that these are actually like endogenous how do we know that these are actually retroviruses? How do we know that it didn't work yet? Because I know that they share the same, like they have a little sequence that looks like a virus, right? So, but how do we know that actually went in instead of the other direction, like where it's like the source of a virus?
So we know for a couple of reasons. One is that we can actually see this process happening in real time. So there's a bunch of situations where we have organisms where they have what are called fixed endogenous retroviruses, fixed ERVs, where that just means that everybody in the group you're looking at, whether it's a population, a species, a broader group, whatever, fixed just means everybody has the same thing at the same place. When that's the case, if you look in the same place in everyone's genome, if they all have the same thing, then whatever that thing is, that is fixed within that group. We can also look at ERVs that are not fixed. Some humans have them, some don't. Some koalas have them, some don't. So what this is showing is the process of endogenization happening. So we have a situation where the virus got in, it got into initially just one member of the population, and then as that member's lineage propagates, we see that virus in some fraction of the population, right? So, and I know for people watching, you may have seen Donnie, Standing for Truth, on here talk about the same topic not that long ago. That was kind of why we're here, because we wanted to. It's fun talking about that topic, and I got name checked a few times in that conversation. So, he agrees, right? And creationists generally will agree that the ones that are not fixed in the population, the ones that are only in some members but not others, those are actual endogenization events. So we all agree that this process of endogenization happens. The disagreement between the evolution community and the creation community is really the extent of it, right? So you have the common young Earth and to some degree intelligent design hypothesis is that the ones that everyone has have some function and they were put there, initially, rather than the result of endogenization. So the evolution community says they're all from viruses. The creation community says some of them are, some of them aren't. And the nice thing about that disagreement is it leads to some very specific testable hypotheses. So we can actually tease apart which one is a better explanation with just a few simple things we can look at, which is a really nice situation to be in. It's one of the few questions that you can really answer in a pretty simple and specific way, which is it's always nice when that's the case.
Okay. Now, what is the evidence that the fixed ones are actually retroviruses?
Right. So that's like the $1 million question, right? It's like, how do we know these things are what we think they are? There's two answers to that. One of the answers is really short, and one of the answers is really long. So we'll start with the really short answer. The really short answer is that there's only one thing on Earth that has this genetic structure and the genetic signature that occurs when it inserts itself into a genome. And that thing is retroviruses. So it has this very specific structure where it's got these things called LTRs, long terminal repeats, on either side. And then it's got three sequences in the middle, the GAG, POL, and ENV. I don't know if you actually pronounce it ENV or it's ENV. I don't know. I always just say ENV, but the three main genes in between. So you've got these two sequences on either side, plus the three main genes in the middle. And then the other aspect to that is the actual process of inserting leads to something called a target site duplication, where the site where the virus actually puts itself gets doubled. So you have a little other duplication on each side of the virus called a target site duplication. You put those things together, the genetic structure plus the target site duplication, the only thing that does that is retroviruses. There's no other, like the target site duplication isn't needed for the virus to do its thing. It's just a consequence of how it inserts itself. So if you find that structure with the target site duplication, you know that's a retrovirus that put itself there. That's the short version.
Okay. Well, I guess, what I've heard about that, and I'd love to hear your response to this, is that we've discovered that those fixed endogenous retroviruses have a very important function in fighting off. It's part of the immune system, and it fights off this process of viruses inserting themselves. So maybe it's necessary that it has the same structure.
So that's so two things with that. One is that, yes, some of them do have a documented function that is an antiviral function. Basically, we have these sequences in our DNA, and if we encounter a similar sequence from an invading virus, then it goes like, wait a minute, that's a problem. Get rid of that thing. The other aspect to that, and the reason that that doesn't preclude endogenization, is that that's something viruses do. Viruses don't like competition. So viruses have ways of keeping out other viruses. And this is not like a retrovirus specific thing. This is lots of viruses do this kind of thing, where they prevent what's called co-infection, which is infection with more than one virus. So HIV does this. First HIV into the cell starts doing its thing. One of the proteins it makes actually degrades the receptors that HIV uses to get into the cell. So like it gets in, says this cell is mine, nobody else is allowed in here. This is my cell. Right? So viruses are actually under selection to prevent other viruses from infecting. Now, why would that persist once they endogenize? Well, that has to do with how endogenous entities, and not just viruses, there are other biological entities like plasmids that operate under similar dynamics. Once you're stuck in your host genome, meaning you can't pop out, you can't become a virus, you can't transmit person to person horizontally. Horizontal transmission is, I have a cold, I sneeze, somebody else gets the cold, person to person. That's horizontal. Vertical is parent to offspring through your genes. Endogenous retroviruses are restricted to vertical transmission. Now, a regular virus, regular old cold virus or a flu virus, right, gets inside you, makes you a little sick, transmits to somebody else. It doesn't really care, right? I'm personifying it. It doesn't care if it makes you sick because it's going to get out and go infect somebody else. That's how it makes its living. Mm-hmm. But if you're stuck transmitting vertically and you make your host sick, that's a really dumb strategy because you're only going to copy yourself when your host has kids. That's the only way you can spread. So you better come up with a way, one, stop making your host sick because that's going to actually end up hurting your own evolutionary fitness, right? You're going to have fewer offspring if your host gets sick. And two, if there's anything you can do to help your host, be healthier, get more resources, have more kids, whatever, if there's anything a virus can do to promote your host doing better, that's going to benefit you as well. So you're actually going to be under selection to help your host. The phrase Donnie used was I think changing sides or something. Well, viruses, once they're vertically transmitted, once they endogenize and become vertically transmitted, they're on your team. They're part of your genome. And that's the only way they're going to replicate. So at that point, yeah, they change sides. They're now, the evolutionary dynamics are going to be to push them to actually help you rather than hurt you. So that explains that activity in the context of the evolutionary dynamics of retroviral endogenization and vertical transmission. That's exactly what we would expect.
Now, but do we have a good reason to believe, I guess, is there evidence of a non-functional endogenous retrovirus becoming functional?
There's lots of evidence of that. The best example is a gene that was acquired in mammals for placental development called syncytin. It's kind of like a stealth mode kind of gene. It suppresses the inflammatory response. So when the embryo starts embedding in the uterine wall, what happens is the maternal tissue sees that as a foreign invader, right? You get inflammation, white blood cells, and that's a problem. Marsupials don't have this protein, which is why their offspring are born so underdeveloped and they have to finish developing in a pouch. They get evicted, basically. And they don't have the time to form a really robust placenta. In eutherian mammals, placental mammals, you have this protein that basically suppresses that response. So you can have a much more robust association between the embryo and the uterine wall, which allows you to have development completely in utero. Now, that's not an ERV that acquires secondary function. That's actually what's called horizontal gene transfer. Basically means we stole a gene from a virus and started doing our own thing with it. The specific question, have we directly observed an ERV that integrates and then acquires a beneficial function in the host? The answer is no. This is a question that SFT loves to ask all the time. And for the life of me, I can't understand why, because it doesn't matter. Function is completely immaterial to the question of common ancestry when it comes to endogenous retroviruses. They could be functional, they could be non-functional. It just doesn't matter. What matters is that pattern that we see in the inheritance. Is it a nested hierarchy across what would be considered created kinds, or is the nesting pattern restricted to within created kinds? And are they uncorrelated across the broader groups? That's the question. Whether those sequences are functional, that doesn't matter when it comes to the question of common ancestry.
Well, I mean, I see how it's extremely important because, if I'm not assuming common ancestry and I'm just seeing similarities, well, similarities are expected with common ancestry, and they're also expected with creation, right? So I expect that, I would have more similarities with a chimp than, some other organism. So like, I guess for me, this question is very important because were these actually the functional ERVs, were they actually incorporated and gained a function, and what evidence do we have for that? That is important.
So that is the perfect segue into that longer answer that I alluded to before. So let's go through the logic of the longer answer. And this is going to take, there's a couple of components to this. We'll go through it little bit by little bit. So the first kind of part of this answer of like, why does the function not matter? Why is it the similarity, the pattern of similarity that actually matters? The first thing that we have to establish, and this is something everybody agrees on, creationist or not, is that these ERV sequences are not constrained. What that means is that the sequence, right? The sequence can change and it doesn't hurt you, right? So these sequences, they can vary, right? We can see mutations in them. And we can see larger scale changes like deletions. I mentioned earlier, they're just often just like missing chunks, right? So instead of having like repeat, gene, repeat, maybe it's repeat, gene, repeat, or maybe it's just repeat, repeat. In some cases, in a lot of cases, it's actually just one of the repeats. It's called a solo LTR, and that's all we have. So these sequences are what we call unconstrained. And we know this. We know that they're unconstrained because we can document, and again, this is not an evolutionary assumption. This is something we can agree on because we can document these differences in them within what young Earth creationists would consider created kinds. So like we could look at apes, non-human apes, for example. We could look at mice and rats and those groups, for example, and we can document differences in their ERVs. So these are things that you and I, even though we're coming from very different directions here, we agree that, all the different mice and the rats, right? Those, I don't want to put words in your mouth, but I'm assuming, I think we agree. I think that's the AIG created kinds has mice and rats together, I think. But you could do it for a bunch of different groups where within the created kind, you can see a lot of variation within the ERVs. And that tells you that you started with something, and then as those created kinds diversified in whatever time frame, mutations occurred, and those things are still around. So there's not a cost to those mutations, right? They're not under selection. Does that make sense to you? And in the chat, does that make sense?
Do we see them becoming functional?
That doesn't. So again, that doesn't matter for the common ancestry argument. What matters is just that they are not sequence constrained, which means they're not under what's called purifying selection. Purifying selection is just natural selection against new mutations or new variants. So if you have a sequence that's really important and has to be like these specific nucleotides, and any difference from any one of them, if that change causes it to not work and it's really important for you to survive, then if it mutates, the individual dies, no kids. So that's strong purifying selection, right? Selection against new variation. What we know about ERVs is that they are not under purifying selection. So the sequences can vary without cost. That's just the kind of the
Okay.
it's the starting point for this chain of reasoning. So they are unconstrained, right? They can change. So with that in mind, with the idea in mind that they're unconstrained, we can make two sets of predictions. We can make predictions based on the separate ancestry model, and we can make predictions based on the common ancestry model. And this is where I will share my screen. So let me here, if you don't mind pulling that up, perfect, thank you. So what we have here are, let's pretend these three phylogenies represent three created kinds, right? So these are separate, dogs, cats, mice, and rats, like whatever it doesn't matter what it is. It's three separate created kinds that we agree within each kind shares common ancestry. So what we should see in a separate creation situation, right, in the separate ancestry model, is you're going to have mutations that occur in each of these lineages, but they're going to be independent of each other, right? Because they don't share a common ancestor. So the changes, so we'll just call this A, right, B, and C, right? So we got three lineages over here. I'm drawing this with a mouse, everybody. So I apologize. This is going to look terrible, but I'm going to do my best. So let's take red. Let's put some red mutations. Let's say a mutation occurs here, right? So then this lineage right here gets a red mutation, right? Those branches get the red mutation. And then that same mutation occurs over here. And then there's a blue mutation. Well, that one's going to occur right here. So this branch gets the blue mutation, and maybe this one over here gets a blue mutation, that branch, right? And then let's take some pink. We'll put a pink, let's say everybody gets a pink mutation over here. This whole tree gets everybody in this tree gets a pink mutation. But then also this branch gets a pink mutation over here. We'll take, let's see, we use a little darker purple. We'll put a purple mutation here, but there's also going to be a purple mutation over here and again over here. Okay. So that's a bunch of mutations. Point is, these mutations are all uncorrelated with each other. And if you were to pick any one of them, you would not get these individual phylogenies, right? Like you say you took the pink one down here, right? You take this branch and group it with this entire tree. Well, that doesn't make any sense. You take the blue one, right? You take our blue mutation, you get this branch grouped with this guy over here. That doesn't make any sense. Those are on separate trees. Because since they're starting from separate common ancestors and they're unconstrained, so they're all mutating, independent of each other, you get an uncorrelated pattern of mutations across these different groups. However, if you look within each group at the specific mutations that we'll find, you're going to be able to recover the nested hierarchy of each group. So within group A over here, you could see that the purple nests within the red. You can see that the pink and the blue are sister groups. They're each other's closest relatives. You'd be able to recover that phylogenetic signal looking just within each kind. But if you went between the kinds, it would be a big jumbled mess. They'd be uncorrelated with each other. That's the prediction from separate ancestry. Okay. Does that make sense? Yeah. Is that okay? Yeah. Cool. Okay, now the opposite prediction, the prediction for common ancestry, is that you can look across A, B, C, D, E, F, G, all, as many groups as you want. And what you're going to find is a nice, neat hierarchical pattern. You're going to find a nested hierarchy where the mutations across all the groups actually do correlate with each other. So that if you have a mutation occurring in one group, you have a good chance of seeing that mutation in the sister group, but you have a very poor chance of seeing that exact same mutation in a very distant group from it. So like, rats and mice might have a specific mutation. Oh, let me look in other rodents and see if they have it. Pretty good shot. Let me look in apes and see if they have it. Not such a good shot because they're more distantly related. So we should see this correlation across groups where we get a nested hierarchy. Instead of being limited to within each group, we should be able to recover a nested hierarchy that spans the entire tree. So we should, I lost my little pointer thingy. Oh, there it is. Here we go. So we should be able to get a nested hierarchy that spans the entire phylogeny and be able to recover a broader pattern if they actually share common ancestry at those higher levels. So that's the opposite prediction, that the nested hierarchy will span across the created kinds instead of being limited to within them.
Okay.
Was that clear? If that wasn't
Yeah.
clear, I apologize. Please ask
No.
questions. Cool. Okay. So now we can take down screen share now. We don't need that anymore. Thank goodness. Sorry for my
Okay.
terrible art, everybody. That's, using the Mac equivalent of MS Paint to do that. So that's what we get. All right. So we have our two competing predictions, very specific, either limited nested hierarchies that are uncorrelated with each other or a universal nested hierarchy. What do we see when we look at endogenous retroviruses? We see a universal nested hierarchy. So within the mammals, we can do this for all the mammals that have endogenous retroviruses. It's not limited just to the mice and the rats or the apes or any other group that we can look at. We can actually see this pattern of similarity and descent that cuts across huge ranges of what would be considered created kinds. And I want to just point out here for everybody, you don't have to assume common ancestry to make this argument, right? We're testing predictions based on the competing underlying model. So we're not assuming one or the other when we're recovering these hierarchies. We're just saying, what pattern do we see? This pattern is compatible with A. This other pattern is compatible with B. And what we find is the pattern that's compatible with universal common descent, not separate created kinds.
Okay, now when you're looking at, let's say you're looking at a mouse, a certain species of mouse, and it has, or you've got an ERV that's common to all mice, right? Mm-hmm. And maybe it's common to some other things, right? But not all mammals have it or something. Then how do you know if that is actually something that has mutated and not just a natural difference? Because essentially at the base level, we're just talking about chemicals that are lined up, right?
Right. We're talking about nucleotide sequences. Yeah.
Right. So how do you tell that this is an actual mutation if you're not assuming common ancestry?
So there's three kind of levels we're looking at when we're looking at ERVs. The first level is just, is there a specific ERV present in a genome somewhere, right? Do you find the sequence somewhere? And they're all going to be a little different from each other. So like within humans, we have the H ERVs. I forget how many complete ERVs we have, but they're all a little different from each other, right? So you can number them 1 through whatever, and we can tell them apart. Or I think it's letters. It's, her A, B, C, whatever. You can tell them apart. So first is just, do you have a specific sequence in your genome? The second is it in the same location in the genome? And then the third thing is, in that location, there's mutations that are going to occur over time. And this leads to what's called SNPs, SNP. That's single nucleotide polymorphism. This is a really fancy way of saying a single base, a single nucleotide in your ATCG letters, just a single one of them is different. That's called a SNP. And so we can look at these three things. Is it present? Is it in the same location? And does it share SNPs? And for each of those three, we get this nested hierarchical pattern. And that matters, and that tells us that it's actually due to mutations in common ancestry rather than being put there in a couple of ways. Kind of each one of those levels addresses part of the concern. In terms of being in the same location in the genome, a common thing that I've heard is, well, they're functional, which they're not all functional. Most of them aren't. Most of them are too degraded to do anything. But let's just say, well, they're functional. So they were put in specific places to do specific jobs. Well, things don't have to be in a particular place in the genome to do a particular job. We know that that's the case. You can look at genes in mammals, for example. We have lots of, we have genes that are shared across all the mammals, but they're in different places. You can even look at the chromosome level, right? You can look at the chromosome level and see different chromosome structure. So we have 46, right? It's our diploid number is 46. We get 23 from mom, 23 from dad. It doesn't have to be that way. There are sister species that have wildly divergent numbers of chromosomes. My favorite example of this is some deer. There's 2 species of muntjacs. These are little deer. And so they're each other's closest relatives genetically. They're unquestionably, the same created kind. But one species has 46 chromosomes and the sister species has 8 chromosomes. So these are very large-scale genomic rearrangements that, again, within the young Earth model and the evolution model, those are things that can happen, right? And in the young Earth model, they happen extremely rapidly because, again, these are really similar species. They're like, you have to be a specialist in deer to be able to tell these things apart. Like, they are basically the same thing. So with these large-scale genomic rearrangements that we know are possible, you can even have smaller ones called translocations, where a little piece moves from one place to another. You can have inversions, where just a little piece flips itself in the genome. That's fairly common. We know that these things happen. We all agree that they happen in the timescales that are relevant to our respective models. So given the fact that we have these rearrangements that occur and knowing the frequency with which they occur, when we find a specific ERV in a specific place, right, that's really strong evidence that that wasn't put there. That's really strong evidence that that was inherited from a common ancestor rather than being put there separately. Because again, in these separate lineages, you're going to get these genomic rearrangements that are going to shuffle things around to the point of even having wildly different numbers of chromosomes, which means you're not going to find the same thing in the same place in the genome. So that's for the kind of the larger scale ERV in the same place. How do we know it's common ancestry and not just put there? And then for the smaller scale questions, the SNPs, how do we know the SNPs are actually mutations and not kind of like created diversity that was front loaded at the beginning? Well, we have to consider that we're going through a bottleneck in the creation model at the ARC where we're only getting a very small number of each species. I shouldn't even say species, of each kind, right? So we can look at the diversity that's present in ERV sequences, and we can see that it exceeds the diversity that would have made it through the flood bottleneck. And then even through recombination, you don't have the rapid enough rate of recombination, assuming you get very different ERV sequences within the two members of a kind that were on the ARC. You don't have the rate of recombination at specific sites sufficient to generate all the diversity we see. So again, the only explanation that works mechanistically is common ancestry and inheritance of these shared mutations over time. And the divergence can be explained by different mutations in different lineages as they diverge from each other.
Okay, well, that was a lot, but I
know that was a lot. I'm sorry. I did two things. I did the big
I'm
scale and the small scale. I'm
not
sorry.
sure that it answered my question because
okay, let's try again.
Because I feel like you're coming from a very strong, like, you know, thing of common ancestry. But when I'm hearing this, and I'm still hearing, you know, like that we don't have a way to tell that these are actually mutations or ERVs. Like I'm still like, to me, I'm still not hearing how we can tell. Now, you mentioned the flood and all of that, but I don't really care about all that. Like, I mean, yes, I'm a young Earth creationist, but my purpose on this channel is really just like examining the evidence for evolution and trying to figure out, is this something plausible? Is this something that should be believed? And so I'm just trying to hear in there, like, how, like, why I should accept that, like, if, you know, these animals have, you know, this particular sequence of chemicals, how do I know that that is an actual, like, virus that made its way in to and became universal and became functional?
So there's kind of two separate questions there. There's how do we know that the mutations, like the little variants that we see that we can use to make our nested hierarchy, how do we know that those are actual mutations that occurred over time versus how they weren't just there to begin with? The answer to that is that we see these variations accumulating in groups of organisms that are with it, like they're a single created kind. So they would have started with, like, if you go back within the young Earth model to the flood bottleneck, the amount of diversity they could have is very limited. And then any variation from that would have had to accumulate since then. So that's the short versions, right? You can only have so many different versions of a site in your genome make it through the flood bottleneck. So the subsequent variation that we see from there that distinguishes, for example, an ERV in this species of mouse versus that species of mouse versus this species of rat, right? They'll share ERVs, but they'll have different mutations that are present, or I won't even say mutations. They'll have different polymorphisms that are present. They'll have different SNPs that are present. How do we know those are mutations? Well, if you've got, you know, an A and a T and a G at a single site, right? Let's work it backwards, right? You could, in theory, at the ARC bottleneck, have all 4 bases, right? Because you got two of each species, right? So you could, and each species has 2, right? Because they're diploid genomes. So you could have 4, right, of each ERV. So in theory, you could have every single base present in the ARC pair at each single site in the ERV. But then you're not going to recognize it as an ERV because you have these 4 completely divergent sequences from each other. And that completely destroys the idea that these are specific sequences that are identifiable by specific hallmarks that are required for whatever function they were created with. So you can only have so much variation. I'm talking about in the young Earth model. You're limited in terms of the amount of variation you're going to start with for these specific sequences, because if they vary too much, they won't look like ERVs anymore. They'll just be like gibberish in your genome. So we can recognize them as ERVs. They're going to have limited variation to start with. So then within all the non-human apes, for example, or within all the, I don't know if old world monkeys are going to be a created kind or new world monkeys, or how specific it's going to get. I don't know what the breakdowns are. But within specific created kinds, within those limited lineages, the variations that we see in those ERV sequences just within those groups, those have to be due to mutations that have occurred since they started diverging from each other. So that's the idea of how we know they're actual mutations rather than created. It's because we can see this widespread variation within what we would consider created kinds. And that allows us to identify the variants that are actually, okay, that variant right there, or this mutation over here, that's a true mutation. And we could work backwards and do what's called coalescence analysis and generate what's called a consensus sequence using the variation that we see in the present and work backwards and figure out what that ancestral sequence would have looked like. And that's important because that gets at the second part of your question, how do we know it's actually from a virus? When we do that, when we work backwards to that consensus sequence, we get an actual functional viral sequence. And this is something that we've actually done experimentally, where you can actually reconstruct an ERV. You could take the ERVs and reconstruct the ancestral virus and show that it works as a virus. Like, it's not just like a virus, it actually works as a virus.
Right. But again, I thought that like a lot of these ERVs are useful in fighting off viruses, and they're like, part of our functioning immune system. So, in one creation model that I've heard, that the idea is that a lot of the viruses we encounter are actually coming from, animal DNA. So this is just, I'm just sharing a hypothesis. Okay.
I have read that too, and I have to say that is one of the most bananas hypotheses for viral origin I've ever heard in my life. It's so let's think about the bank shot for that, for how the creation model would work if viruses are actually derived from endogenous retroviruses. The logic there is that at creation, our genomes were loaded up with these ERV sequences that don't match to anything in the environment. They don't match anything in the environment because viruses came from them. So they were put in our genomes to defend against something that doesn't exist. And the thing that they defend against actually came from the sequences that were put in our genomes. That's, I mean, I don't like cover theology on my channel, but that makes no sense. Right?
Okay. All right. You're making a decent point, but it could be foresight. You don't know. But look, I mean, it could be. But
it could look, that gets again idea, theology and like, did you know they were going to sin and everything? Right. It gets, it gets sticky and I don't cover it. Yeah.
But what I'm trying to say is, like, I don't yet see a reason to accept what you're saying. I don't see a reason that I should accept this idea that these fixed ERVs are actually viruses that made their way into our genome and now have a function that helps us. I mean, that equally seems kind of bananas.
So right? I will just say two things there. One, when we say they have a function, that is, they make up about 8% of our genome. The vast majority of them are degraded to the point where they aren't doing much of anything. They're either just the paired LTRs or a solo LTR, and all the stuff in the middle is missing. To the extent that they have the stuff that's present, it's often highly mutagenized, which would undermine the function of, like, my sequence, the ERV sequence, is going to match an invading virus. If they've accumulated a bunch of mutations over time, because again, they're not constrained, they're not under purifying selection. So if they've accumulated a bunch of mutations over time without any apparent fitness cause to it, they're not going to do the job of matching something that's going to invade you from out in the environment. They're too different. The other side of that is ERV sequences in the human genome are often what's called, they're methylated. So this is a form of regulation where you chemically modify the DNA with methyl groups. This is important because methyl groups suppress any kind of activity. They basically cause the DNA strand, instead of being kind of strung out like this, they cause it to clump up and kind of knot really densely. And when you have a heavily methylated sequence that's really tightly wound up like that, that's called heterochromatin. And heterochromatin isn't expressed into RNA or anything. It doesn't do anything, for the simple reason that there's no room for the enzymes to, like, get in there and grab on, because of this kind of dense coil of DNA, right? The enzymes need space to, like, you need the DNA strand, and the enzyme kind of grabs onto it and does its thing. If the DNA is all coiled up really tightly, the enzymes can't do that. So any region of the genome that's going to be heavily methylated, which is a large portion of the genome, and ERVs are largely methylated and therefore biochemically inactive, they're not doing anything. They're not doing, let's make some mRNA and then let's make some RNA and then the RNA can go match to a virus and we can kill the virus because it matches. You can't do any of that with a sequence that's heavily methylated. So between those two things, either degraded to the point where they're not going to be useful, like as an immune kind of defense situation, and heavily methylated to the point where they're biochemically largely inert within the cells. You put those two things together, that's the vast majority of ERV sequences, which, if you ask me, is extremely strong evidence that they're not doing anything functional, especially pairing that with the fact that the mutations just accumulate, the single base mutations and the deletions. If they were doing something important, you can't just, like, delete parts of them. But out of the 200,000 that we have, like, over 199,000 of them have deletions of some kind. So, like, they're probably not doing anything.
Okay, so are you saying we understand the non-functionability, like we understand the, like, what's functional and not functional in every ERV in?
Oh, no, absolutely not. No, I think so. We have a really good picture for a lot of them. I'll go out on a limb and say, I think if it's a solo LTR, that thing's degraded, is not doing anything. Probably the LTRs with no genes in the middle, maybe they're doing something, probably not. And then if it's heavily methylated, you could probably take that to the bank, because if it's heavily methylated, it's biochemically just not active. That's what that does. It shuts it down. And it's hypothesized that that's actually an antiviral defense that our cells have, that we identify viral sequences and methylate it so they don't cause problems.
Well, I think, SFT's response to this was, first of all, he was saying, us trying to figure out what functions are happening with the DNA is like me, I don't know if he said, do you speak Russian? And I don't. So he said, but you might, I don't know. Do you speak Russian?
No.
Okay.
No, I did watch the stream, and I remember that part. Yeah.
Yeah. So he said, it's like as if you've been given, you're supposed to proofread, a giant Russian document. Are you qualified to do that?
I think a better analogy is that we have Egyptian hieroglyphics and we have a Rosetta Stone, and we're figuring it out as we go, right? We have a partial key and we're figuring out more. It turns out there's more pieces of the Rosetta Stone. We keep finding more that help us understand this biochemical stuff that's going on. I think that's a much better analogy. It's not, if it's me or you editing Russian, I have no experience. Like, I couldn't even read the alphabet. I don't even know what all the letters are, right? Because it's a different alphabet. I got nothing. But that's not how it is with the DNA. You know, we sequenced the first human genome about 20 years ago, give or take. We sequenced the first genome entirely in the 70s. It was a tiny little bacteriophage. The first DNA gene, that was an RNA, was first, then DNA, another tiny little bacteriophage back in the 70s. And, you know, so we've been at this for a while now, and we've gotten a lot better at it. So there's unquestionably stuff that when we first looked at it, you know, in the early 2000s, we said, that's not doing anything. And we've since figured out what it's doing. There's unquestionably stuff like that. But for every instance of that, there's other stuff where we've looked into it, we've done the tests and said, yeah, that's not doing anything. Right. And it's not that we're starting from, okay, we understand the function in 5% of the genome, and we don't know the other 95%. And now we understand the function in 8% of the genome, and we're still working on the other 92%. It's as the functional part of the genome has gone from, 3% to 5% to 8% to like now, if you stretch it a little bit, you can get 10, maybe 12%, like really good evidence of function that is preserved through natural selection. You get into the very low double digits. As we've increased that fraction from 3% up to like 10, the rest of the genome, it's not still some unknown black box. We've figured out what a lot of it is, what it's doing, and just as importantly, what it's not doing. So it's not this giant, 20 years ago, if you want to argue the genome is entirely functional, I think you have like the ability to say, well, we have to look, and it might end up being more functional than we think. Now, we're really sure about the endogenous retroviruses, pseudogenes, and the various classes of retrotransposons. Like, we're really good at documenting what these things are doing and how they're operating and where they're operating, the levels at which they're operating. So we have a really strong case now to say huge swaths, 50, 60, 70% of the genome, it's doing stuff, like biochemical stuff, but it's just kind of stuff. It's not helping us in any way.
Okay, well, we got so far off. I don't remember what the other response that Donnie said about.
I wrote them down. I wrote them down because I figured they might come up. So I actually went down and wrote them down. So the big thing, talking about this hierarchical pattern, the big objection was related to hotspots. Was this kind of, it was a multi-step thing. But if Donnie, if you're watching, correct me in the comments if I'm, if I get any of this wrong. But I think I'm going to present this fairly. And I'll also say, by the way, I did reach out to Donnie to have this conversation. Rebekah, I think you're a very fair person. If you would, but I'm sure there are other places that would host Donnie and I. He initially said yes, and has since kind of ghosted me. So I don't know if that's going to happen, but for everybody who's like, why aren't they just talking to each other? I have the same question, and I hope we do get to talk to each other at some point in the near future. So cool. The idea here is that, okay, you can invoke these things called hotspots to explain this pattern. Hotspots are regions of the genome where you have an elevated mutation rate. So we've known for a long time that mutations are not uniform across the genome, both the types of mutations that occur and the rate at which they occur. There's hot spots and there's cold spots, right? Where you get a lot of mutations, you get very few mutations. So what Donnie said was, well, these regions, they could be hot spots, and that would explain the convergence of the different mutations across these different lineages, across these different created kinds. They're hot spots, they're going to get the same mutations. So the first problem here is that if they were functional in the way Donnie and other people describe them, that would require some degree of sequence specificity. So they wouldn't be hotspots. They would you'd be under they'd be under purifying selection. But that's a very small objection. The bigger problem is that hotspots just mean that mutations are faster. It doesn't mean that specific mutations are more likely or going all the way are predetermined, right? And this is not like a new finding that mutations are, you could use different words for it, random. I don't like random as much because you say random and people think it's a coin flip, right? Think it's equally likely, and that's wrong. But some good words for mutations are probabilistic and stochastic, meaning they just happen. There's a probability distribution, there's a pattern to them, but they're not deterministic. They're not specific mutations are going to happen in a specific order, right? So within that probability distribution of mutations that might happen, hotspots make them happen faster, but they're not going to dictate which specific ones happen, nor are they going to dictate the order in which they happen. And both of those things matter. If you find a nested hierarchy that spans like all of mammals, for example, you need to explain not just why those mutations are shared across the lineages, but you have to explain the order that they occurred throughout history to explain the specific nested pattern, right? The box in a box pattern that we see. You need, again, the same mutations to happen in the same sequence, going back through these different independent lineages. So in other words, what you need are the mutations to be deterministic rather than probabilistic. And that was disproven in an experiment in 1943. Like this is not like something that's up for debate. Mutations are not deterministic. It's just, they're not. So that invoking hotspots to explain this pattern doesn't work. And that's why.
Okay. Well, what would be the reason to accept the idea? Because you kind of talked about it in terms of like if we were looking at created kinds, right? But of course, for you, it goes back,
way back,
right. So, but what would be the reason to accept that it goes back? And so what I'm saying is like if you're looking at an ERV that, let's say it spans like all mammals or something like that, why would that necessarily be an endogenous retrovirus that made its way in and then spread to all the mammals, rather than in, my understanding that, God created with certain features and those happen to be similar features within all those organisms. So what would be the reason that I would accept that it went all the way back to where the common
I want to put a pin in that just for a second, because I want to be fair to Donnie here, because I did not give his full and complete argument. There was kind of a second layer to it, and I don't want to leave that out in fairness. I don't want to address the first part without covering. I don't want to shortchange him here. So the second kind of layer to this, say, okay, mutations are probabilistic rather than deterministic, but we can look at certain parts of the genome and we see what's called convergent evolution, where the same thing happens in different lineages, right? And this is something that is agreed upon, again, in both models, right? So we can look, there's a really famous one is a pseudogene that's involved in vitamin C synthesis. Like we can't make our own vitamin C. It's why we get scurvy if we don't get vitamin C. We can't make it ourselves. Lots of mammals can make vitamin C. Lots of mammals can't. They have this one gene, it's called the GULO pseudogene, GULO. It's involved in vitamin C synthesis and it's broken in a lot of different mammal lineages. So you say, okay, well, if you can have convergent evolution like that across all these different groups that like evolutionist, right? I'm not saying everyone shares a common ancestor for that broken GULO pseudogene. That broke independently in a bunch of different lineages all on their own, right? So like that's called convergent evolution. And then even more specifically than that, you could have what's called parallel evolution, where the same mutations occur in different lineages over time. We see this in like antibiotic resistance. So like in MRSA, for example, you keep seeing the same resistance mutations evolving in separate bacterial populations, like the exact same mutations over and over again. So like, why can't that be the case with herbs? And the answer is twofold. One is you need one of two things to happen in order for those explanations to work. One is you need really strong selection for a specific trait, like antibiotic resistance. Like if those bacteria don't get resistant, they're dead. So you have really strong selection, and you have situations where there is an optimal solution to the problem, right? There's an optimal genotype you can find that makes you resistant. So that's one way to accomplish this type of parallel evolution, is you have a really super strong selective pressure and a limited set of options. That's what we see with antibiotic resistance. The other thing that we see that leads to this situation is when you have what's called relaxed selection. It's kind of the opposite, is where instead of having really strong selection, you have no selection. It just doesn't matter. So like if you're an animal that eats plenty of vitamin C in your diet because you eat like fruit, for example, maybe You just get plenty of vitamin C. So if your vitamin C gene breaks, who cares? You'll be fine. You eat fruit all the time. Like, that's your diet. So it doesn't matter. So that gene is under relaxed selection. If it mutates, if it breaks, it activates, who cares? So what we see is, in that example with the gulo pseudogene with vitamin C, it tends to be broken in the lineages that historically eat plenty of vitamin C in their diet. So there's no cost to not making it. In the lineages that don't get vitamin C in their diet, you better have that gene working, because if you don't, you're going to get scurvy and die. So that's the other option, is you can have relaxed selection. But what happens in that situation is you don't see the same mutations in all these broken pseudogenes. You actually see different mutations in each lineage that lost it. You see the same types of things. You often see deletions, but you don't see the exact same things happening over and over again. Neither of those explanations apply to ERVs. They're under relaxed selection. They're not under purifying selection. But you see the same mutations in this nested hierarchical pattern. So neither one of those potential explanations for this convergence explanation actually works to explain the pattern that we see in the endogenous retrovirus. So I apologize. I know it was long, but I wanted to give that argument fair attention. I didn't want to shortchange it.
I guess when you're talking about this vitamin C thing, you called it convergent evolution. But if it is what you said it is, and these are broken, then it sounds like convergent devolution. But
devolution.
It's a ruining of something rather than a building up, which is what we need for evolution to happen. So,
but that's different from the pattern of common ancestry. That's independent of the common ancestry argument.
Right. But I guess what Donnie's response to that was that we don't, we don't know that gulo, whatever.
Yeah, we do. It doesn't even have a promoter. It's broke. Okay. It doesn't even have a promoter. It doesn't do anything. It's super broke. We do know.
Okay.
Yeah, we know. It's not that thing is super dead. It doesn't do anything. It doesn't have a function. Like, it's dead. Yeah. This would also be really, Donnie, this would be easier if we could just do it ourselves instead of having Rebekah here, who is great at this, but like having her relay the arguments back and forth, because when you were here, she was like, oh, I watched Dr. Dan's video. What about this? And now we're here. I'm going, yeah, I took notes. And she's like, oh, but he said this. It's like, this would be so much easier. Just talk to me. This would be so much easier.
Yeah, it would. I hope we can do that. And I would love to host that on my channel. That would be great. Okay,
we've always been nice when we talk to each other in person. We're always nice.
Yeah. And I appreciate that you're willing to take the time and explain all of this.
I'm happy to. I love this stuff. This is great.
Yeah. Is there anything else that you want me to understand about ERVs? Do you think I'm getting the point here?
I think you are. And I want to go back to the question that I said I would come back to, and I apologize because we're on that second part of that argument, but I want to come back to the question. The question is, what's the reason to accept common ancestry for ERVs? Like, if all mammals share a specific ERV, you specifically said, what is the reason that you would accept that would like get the point across that it's common ancestry? I think the answer to that question is there isn't one. I legitimately don't think there's anything I could say, or any paper I could show you, or any data I could show you. I don't think there's anything I could say for you to accept the common ancestry of all of mammals, for example, based on endogenous retroviruses. But that does not detract from the fact that we have a nice, neat case of testable hypotheses here, right? We have two separate hypotheses, common ancestry, separate ancestry, and we have two distinct sets of predictions that flow from those. Can you get a nested hierarchy within kinds, but uncorrelated between them? Versus, do you get a nested hierarchy that's correlated across all of the different kinds, right? And when we look at the data, what we see is the latter, which is extremely strong confirmation of the common ancestry model, and notably direct refutation of the separate ancestry model. I don't think that's going to convince you.
What do you, give me a reasonable, like, what is the thing that is like against separate ancestry? Like, so what are you seeing that is common to, all these organisms?
Because if we have separate ancestry, These sequences are unconstrained, so they accumulate mutations, right? They're not locked in place. They can freely accumulate mutations. We know that's the case. And mutations are stochastic. They're probabilistic. So we don't know what specific mutation is going to occur in a specific place in the genome. We don't even know what type of mutation. Is it going to be a single base, an A changes to a G, or is there going to be a region that gets deleted somehow? These things are all probabilistic. So when we see the same pattern of specific mutations within ERVs in this nested hierarchy across kinds, that's something that is explicitly contrary to what you would predict in the separate ancestry model. Because in the separate ancestry model, you've got a starting point over here, you've got a separate starting point over here. They're going to mutate subsequently to their creation. Those mutations are not going to have anything to do with each other. They shouldn't be correlated with each other. So if we see a specific, A to T mutation over here, there's no reason to expect that same mutation over here. If we see a specific deletion over here, there's no reason to expect that same deletion over here. But we do see the correlation of these mutations, again, in this nested pattern, which is explicitly contradictory with that separate ancestry model.
But I mean, you just gave a reason. You said that, like with the vitamin C one, that we have species that have it functioning and species that don't have it functioning. And it's not from a specific nested hierarchy, right? We can't follow, we can't trace it
in each one, right? And the reason we know it's independent in each one is that the mutations in each one are distinct from each other. It would be really confusing if everybody had, because if you were to do like a phylogeny of all mammals, right? Like all the mammals, you would have like this branch and then this branch and then this group over here. You'd have a bunch of like seemingly random groups just shotgun throughout that phylogeny that have this broken gene. If all of those different lineages had the exact same mutation that broke that gene, man, that would be really confusing, because that would imply a common ancestor for that pseudogene when all of those things shared a common ancestor, if you go with the common ancestry model. But that's not at all a parsimonious explanation, because then you have to have it get fixed and all these other, and when I shouldn't say use fixed, you have to have it repaired then in all these other lineages who've descended from the common ancestor where the original mutation that broke it took place. Like, it would be a big giant mess. There's no reason to think, like, in that case, that would be super confusing. And I don't think common ancestry would be necessarily a good example for that if it was the same mutation, because that would not follow any kind of nice nested pattern that we'd expect to see. Well, that's not what we see. What we see is different mutations that inactivate that gene in the various lineages where it's broken into a pseudogene. We don't see the same mutation over and over again. We see different mutations in the various groups, which is what you'd expect for something that happened independently many different times. Okay.
Well, I guess I'm kind of asking for a specific, what is one ERV that would point back to common ancestry?
That's a great question. It's not any one ERV that does it. It really is all about this pattern. So, for example, just looking at the great apes, right? You get humans, chimps, gorillas, orangutans, and in that order, right? So humans and chimps are sister groups, and then gorillas are the next closely related, and then orangutans are the next closely related. Now, I know the numbers for humans and chimps, where out of about 200,000 ERV sequences, of which like 1 to 2 dozen are like the complete ERVs, so we share all of those. I think we share all of them. And then out of the 200,000, the vast majority are some form of degraded. We share over 199,000 of them. So very similar in terms of the ERVs in those two genomes. Now, if you go up to gorillas, you see that they have more that are on their own and fewer that are shared with humans and chimps. If you look at orangutans, again, they have more that are limited to the orangutan lineage compared to their similarity with the gorilla and the human and the chimp lineage. So they have less in common with humans and chimps, even compared to gorillas, right? So again, you get this nested hierarchical pattern where you get less in common as you go further up in your level of diversity that you're looking at. So it's not looking at any one specific sequence and kind of tracing its evolution through all these groups. It's the pattern that forms when you look at them as a whole, and you look at the degree of similarity between things that by other measures we say, okay, they're closely related, therefore we can predict they share X percentage of their ERVs. And then you look at the kind of a group that's less closely related, say, okay, it's going to be Y, which is less than X percent. It's going to be Y percent. It's 5% less, whatever it is, right? And we can make these predictions and then verify that they do actually form the pattern we expect. So it's not tracing one specific sequence through its evolutionary history. It's the pattern that we see across these diverse sequences in diverse lineages, and the correlation or lack of correlation between them is what really brings out the common ancestry.
Okay, but I mean, you understand that from my perspective, it's of course a gorilla is going to be closer, right, in similarity, and so I mean,
I hear you, but
yeah,
no, I hear what you're saying. It's just that that's a testable hypothesis, and we can evaluate within those created kinds, right? So I think, if I have it right, I think all the other great apes are a single created kind. Orangutans, gorillas, chimps, bonobos. I think those are all a single created kind by most models. So what we would expect then is to see all of them be more similar to each other because they're a created kind, right? And then humans are a separate created kind. Homo sapiens are a separate kind over here. We would expect them to be more similar to each other because they, in reality, share a common ancestor, whereas they do not share a common ancestor with us. Even if that ape common ancestor was created very similar to the Homo sapiens, right, to Adam and Eve, if they were created very similar, that ape common ancestor the descendants of that ape common ancestor should be more similar to each other than any of them are to us. But that's not what we see.
Okay.
In
what way? With the ERVs or with mutations?
With the ERVs, yeah.
Okay.
Yeah, with the ERVs. Because again, these are unconstrained sequences. They're just mutating, I won't say randomly, but they're mutating probabilistically. So any of the ERVs that are created in this ape common ancestor, the pattern that we see in chimps, gorillas, orangutans in the modern world, those things should all be more similar to each other than any of them are to the ERV pattern in Homo sapiens, because we don't, in actuality, share a common ancestor. So no matter how closely similar they were made at creation, just because, randomish mutations occur, there's no reason that chimps should mutate in a specific way that makes them more similar to us, while the gorillas and orangutans become more different from us over that same amount of time. There's no mechanism for that. There's no reason for that to happen in the model, in the separate ancestry model. But the common ancestry model exactly explains that pattern. That's the idea I'm trying to get across.
Okay, I'm not sure I get it. I might have to go back and listen to this a few times. So,
okay. I'm sorry. I know it's a lot.
It's a little overwhelming. Well, I guess, but maybe the point I'm trying to get at and trying to understand here is that, so the vitamin C got broken like that. Okay, it got broken separately, right? It got mutated.
Multiple different lineages, yeah.
Okay, and so in that case, that's not an example for common ancestry, right?
Right. So you can look at each instance of breaking and do kind of a limited common ancestry within each example. But across all the different ones, you don't see the correlation there. Yeah, exactly. And we can do that. We can be sure of that because two reasons. One is that the mutations are just different, right? You can compare one set, one lineage of gulo pseudogenes to a different one, and you're going to see that the mutations that inactivate them are different. The other reason is that we can look at other sites in the genome and say, okay, do we expect them to have shared based on when this mutation would have happened or this inactivation would have happened? Do we expect them to share a common ancestor with that? Or would the common ancestor be too far in the past for that to have made sense? Would there be other lineages in there that don't have the pseudogene? So it seems, I feel like it seems contradictory that I'm saying in one case, yeah, we have this nice, neat nested hierarchy, common ancestry. In the other case, we have not a nested hierarchy. We have convergent evolution, right? We get the same phenotype, broken gulo pseudogene, but we get it through different ways, not the same mutations. And we say not common ancestry, and that's all consistent. The reason that's consistent is because in both cases, what we actually find in the world is the prediction made by the common ancestry model based on what we see in other parts of the genome. So we can say, based on other parts of the genome, humans and chimps are the most similar to each other. Therefore, they should be the most similar when it comes to ERVs. And we could do that,
right. But if
scale, large scale.
If by if only the unfixed verbs ERVs, only the unfixed ERVs,
I know what you mean, yeah.
Okay, are actually ERVs, like endogenous retroviruses, and the fixed ones are actually something that were put there by God, right? Okay, I'm totally forgetting my point here. But what I'm saying is, then I would expect to see a similar thing put there by God in chimps to humans because we are more similar.
But here's I hear you, but here's the important thing. At least I think it's AIG's model has all the apes being a created kind, right? Apes, chimps, chimps, gorillas, orangutans. Meaning they share a common ancestor, not just at creation, but they share a common ancestor pair from the ark, right? So those three species would have it's actually for bonobos and there's other, whatever. Though all of those species, right, the non-human great apes would have shared a common ancestor. So there's no reason that one of them would be more similar to humans than any of the others, right? They're all equally related, which is to say they're not. They're related to each other, but not to us in actuality. So you can't say, well, they created chimps to be more similar. Well, chimps wouldn't have been the created type. It would have been the common ancestor of chimps, gorillas, orangutans. So the question stands, why are chimps more similar and not any of the other descendants? And why does it form this pattern that we see?
Right, but that's assuming AIG's model, which could very well be.
Oh, you could do it with a different model, sure. Sure. We could use a different model with more or fewer created kinds, but at some point in the model, you're going to run up against that exact problem. Because the nesting, no matter where you draw your lines in the model, the nesting crosses the lines. So at some point, no matter where you draw the lines, you run up against the problem I just described. It may not be for humans and chimps and gorillas and orangutans, but it's at some point you get to like, okay, if this thing, why is it more similar to this thing than this other thing if they don't actually share common ancestors, right? That's what you run up against.
Okay. I think Andrew Cumming had a question for you. Andrew, please put it in the chat again because it may be lost. And I appreciate the audience being here. But while you're getting ready to put up your question, I wanted to know, Dr. Dan, is there anything that we didn't cover that you feel like should be covered? Like, did you hit the major points?
I think we hit the major points in terms of the affirmative arguments for common ancestry for ERVs and the hypothesis testing of the separate versus the common ancestry model. I will tell you, I watched with great interest your chat with Donnie from last week, and I took lots of notes on it, and I have several pages of corrections, but we'll not go through those because we'll be here all night, so we won't do that. But in terms of I think we hit the high notes.
Okay, well, then we'll try to get Donnie on for a discussion and hopefully you guys can, talk about it because obviously I don't have the expertise in this area. So I don't really know, what the response is, should be. So, but sorry.
So I have, where is it? Where's the book? I have the book right here. Okay. I have this. This is what started this whole thing. I talked to Charles Jackson a few months ago. We ended up spending like 3/4 of our conversation talking about ERVs. Donnie didn't like that. He had a long back and forth with Dr. Jackson. Then I was like, let's talk about ERVs. And he's like, no, I don't have time to talk about ERVs. And then he wrote a book about ERVs. And someone bought the book for me. So then I reviewed the book about ERVs. And now we're having this like game of telephone about ERVs. And I will say there's a lot of problems with that. You saw the review. There's a lot of problems with that book. There's a lot of problems. So a lot of problems with that book.
Okay,
Donnie,
well,
here's
talk. Andrew's question, and if we have time, I'd like to ask Paleo Logos. He has a question too. But so he says, can he please give his thoughts on the idea that we know these differences are from mutations due to the probabilistic biases in their occurrence, transitions versus transversions?
Yeah, that's a really good question. So a little bit of background here, and then I also see Paleologos question. Hi, thanks for coming. And then I also hand waving the topic of function. Yeah, I got my fan club is here. So so the idea that so this goes back to the idea that not all mutations are equally likely. And that actually informs the models that we use when we are inferring common ancestry or evaluating relatedness, right? So we can say, for example, what Andrew says, transitions versus transversions. So there's two types of nucleotides. There's big ones called purines, and then there's small ones called pyrimidines. And within the DNA, you've always got a purine with a pyrimidine. So it's always a big with a small. And that's why the helix stays a constant diameter. You never have big with big or small with small. So when a big nucleotide, like an adenine, changes to the other big one, a guanine, that's called a transition. When a purine changes to a pyrimidine, or a pyrimidine changes to a purine, that's called a transversion. And transversions are much less common. So if you have, for example, an A to G transition, there's like, if two things have this exact same transition in the same place, there's like not a great chance, but like definitely not a zero chance that that's convergent evolution. If you have two things that have a specific transversion or a set of transversions that are all the same, so like A to T, C to T, or sorry, that's a transition, G to C, right? You have a bunch of transversions and they all match. That's really strong evidence of common ancestry because transversions as a type of mutation are relatively rare. So when you have several rare events that all line up, that indicates that they're common ancestors. So you can look at the type of mutation that's occurring in addition to what it specifically is, right? Is it this specific deletion, this specific single base substitution, whatever it is? You can evaluate the frequency of that class of mutation, and that can inform how you evaluate your phylogenies. So that's what Andrew's talking about.
Is your assumption that it's a mutation based on the idea of common ancestry? Like what I mean is like, are you lining up these genomes and being like, well, we know they had a common ancestor and therefore we know this P or this, G doesn't fit here.
So actually, no. So what you're doing when you do that type of math is you're explicitly trying to evaluate if you can assign a specific mutation as being due to common ancestry or not. You're actually trying to distinguish between common ancestry and not common ancestry in that situation. So if you have like a specific mutation that for whatever reason is really super common, like a C to T mutations happen super all the time. They're just, for whatever reason, C to T mutations. We don't have to get into the reason because it's, complicated, but C to T happens all the time. So if you have one specific, you have a long sequence, right? And you've got a specific C to T mutation in there, like that's not super strong evidence that they like that if they share that C to T, that's not super strong evidence of common ancestry because C to T happens very quickly. That's a very common type of mutation. Whereas if you have like an A to C mutation, that's very rare. So that would result in a stronger assessment of common ancestry if you have a specific A to C mutation that is shared, or if you have a deletion that starts and ends at exactly the same point, right? That would lead you to be more confident in your analysis of common ancestry, right? That's the idea, is that you're not assuming common ancestry when you consider those things. You're actually using those differences in frequency to evaluate common ancestry. Yeah, that's a cool question.
Yeah. Okay, so can you ask Dr. Dan about the probability that viruses would become attached at the same regions of the genome when infecting different hosts?
Yeah, so this is a common thing that I haven't heard this too much lately, because what I've been dealing with is a lot of the idea that ERVs are actually not from viruses at all. But in the kind of side of creationism, where they accept that ERVs are actually derived from viruses, what the argument is to explain the nested hierarchical pattern is that, well, they have preferential insertion sites where they target, right? So it's not random that they inserted in all these different places and they just happen to match across these broad lineages. It's that, well, the viruses are going to specifically target these particular regions. Now, there is, like many things in creation science, a nugget of accurate information that is built on. And the accurate information that this idea is built on is that there are hotspots and cold spots for viral insertion. Different viruses tend to insert preferentially in different parts of the genome. It's not a like, I'm going to hone in on this specific part of the genome, but it's regions where they can kind of target. And sometimes it has to do with like being AT rich versus GC rich, for example, or it'll be a region that tends to be, it'll be near expressed genes because the genes are where the DNA is unwound. Whereas in the areas that we talked about before, if they're inactive, if they're methylated, then the DNA is all crunched up. So there's not the, ability to grab onto that DNA and insert yourself. So you do see preferential insertion in specific places in the genome. And we can actually see this in viruses that cause cancer. We can see the hallmarks of this. So for example, HPV, human papillomavirus, causes cervical cancer. That's because some percent of the time it inserts in the vicinity of a gene that is highly implicated in cervical cancer. HIV, one of the signs of HIV early in the AIDS pandemic was a specific type of skin cancer called Kaposi's sarcoma. Same idea. Not 100% of the time, not even a majority of the time, but some fraction of the time when that virus inserted, it tended to insert near some genes that were implicated in this type of cancer. So you tend to overexpress or break the regulation or whatever of those genes, and that leads to the cancer. So there are hotspots and cold spots for viral insertion, but like mutations as a whole, they're probabilistic. They're not deterministic.
Okay, well, thank you so much, Dr. Dan. It's been great having you on. And for those watching, Dr. Dan's channel is Creation Myths, and he does a lot of talking about this topic. So it was great having you on, Dr. Dan, and thanks everybody for your great questions and comments, and good night.
Thank you, everybody. Talk to me, Donnie.